At the end of our previous video focused on gastrin, the primary regulatory hormone of the stomach, we introduced the name of the second regulatory hormone called secretin that directly counteracts the effects of gastrin and therefore prevents the stomach from doing damage to itself or to the duodenum and all of the digestive organs that follow it. This is because secretin is the hormone that is directly responsible for triggering the release of sodium hydrogen carbonate by the pancreas in order to prevent any damage to the duodenum and the rest of the small intestines. Now, as when discussing any regulatory hormone, the first place that we need to start is the stimulus, which is the change in environmental factors that leads to the production of secretin. And this occurs when the stomach digestion is done, because when the stomach has basically finished digesting all of the proteins that exist, all of the chyme, which contains HCl, the acid that activated pepsinogen and that's used to denature any proteins within your food, all of this would be pumped out of the stomach into the duodenum. Now, the problem with the duodenum is that because the duodenum is part of the small intestines and therefore is used for nutrient absorption, the walls of the duodenum lack the protective structures that are found on the walls of the stomach, and therefore if the pH is extremely low due to the presence of HCl in the chyme from the stomach, this can cause very severe damage to the walls of the duodenum. And consequently, when a high concentration of HCl is detected by cells of the duodenum, that signals these cells to produce our second regulatory hormone, secretin, and the primary location where secretin is going to travel after being released into the duodenum is actually back up to the pancreas. And this is because the pancreas is the location where sodium hydrogen carbonate, or NaHCO3, which functions as a weak base, this is where our weak base is produced. And then once the pancreas has produced a sufficient amount of sodium hydrogen carbonate, it transfers this sodium hydrogen carbonate into the duodenum in order to neutralize the HCl from the stomach and therefore prevent the acid from doing any permanent damage to the walls of the duodenum itself. Now, secretin doesn't actually function on its own. There is a second regulatory hormone that upregulates the pancreas in terms of signaling digestive enzyme production, but the other function that secretin actually has on the pancreas is dealing with high concentrations of glucose, which of course is a sugar produced by the breakdown of starch that actually occurs in the mouth. And as we learned when we studied the pancreas, the hormone insulin is the hormone that tells the cells of your liver and of your muscles to absorb this high concentration of sugar in the blood, transfer it into storage, and store it as glycogen. And one of these side effects that doesn't really relate to the regulation of pH is that secretin also triggers the pancreas to produce insulin, although once again, this is not really its primary function. Now, directly related to its primary function, secretin, in addition to traveling to the pancreas, can actually travel up to the stomach itself and stop the stomach from actually producing gastrin, which means if there is no gastrin, that means that no acid is going to be pumped into the stomach, and if no acid is pumped into the stomach, that means that not only is the enzyme pepsinogen not going to be activated, meaning that it won't be converted into pepsin and therefore won't be able to digest the protein in the muscular walls of the stomach, but it also prevents HCl from ending up in the duodenum, thereby preventing any damage due to acid from being suffered by the walls of the duodenum, and secretin cuts this off directly at the source, that being the cells that pump HCl into the stomach. Finally, we also need to make sure that we have a way to tell the pancreas that it no longer needs to produce NaHCO3, the base. So when the hydrochloric acid from the stomach has been successfully neutralized, if there is any sodium hydrogen carbonate left over, this would result in the pH of the duodenum actually becoming basic rather than acidic, because if there is no stomach acid to neutralize this base, it will turn the pH of the duodenum neutral, or past neutral rather, becoming basic. At this point, this is the shutoff signal that stops the cells of the duodenum from producing secretin, because they recognize not only is the acid gone, but we actually have too much base in this area, and therefore that the sodium hydrogen carbonate is no longer necessary, and therefore this shutoff signal is sent to the pancreas, meaning that no secretin ever makes it to the pancreas, and therefore NaHCO3 release is inhibited, which is also another example of feedback inhibition, because the more NaHCO3 makes it into the duodenum, the more likely it will neutralize the HCl, thereby reducing the need for base to be pumped into the duodenum, thereby shutting off the entire system before excess base can accumulate in the duodenum. The final regulatory hormone of the digestive enzyme is actually a companion hormone to secretin, because once again, the other primary function of the pancreas is to produce the primary digestive enzymes that are used to break down undigested food after the stomach. And in the final video in this series, we are going to introduce that hormone and the effects that that hormone has on digestion of the duodenum. And so we will introduce the short form of that hormone, which is a hormone that we call CCK, which we will focus on in the final video in this series.